Okay, hello students, welcome to Affinity Digital. Myself Usha. I hope you have done your questions well. So the mini static test number 7. So let's discuss the questions. So let's start with the question. Okay. So the first question is energy required for ATP synthesis in PS2 comes from. In the process of photosynthesis, the ATP synthesis comes from proton gradient. It should be from proton gradient. Okay. Next is the correct sequence of flow of electrons in the light reaction is. So this is about the ETS. So in the ETS, the sequence of the electrons in the light reaction, this will be PS2 plus toquinone, cytochrome PS1, and then pyridoxine. Start from PS2, then plus toquinone, then cytochrome PS1 and pyridoxine. Okay, chlorophyll A is the chief pigment associated with photosynthesis. This is correct. The main center is occupied by chlorophyll A. Second, the reasoning is in the blue and the red region of spectrum, there is maximum absorption by chlorophyll A and also shows higher rate of photosynthesis. This is also correct because in the blue and the red region, it comes under PAR. PAR means photosynthetically active radiation. This comes around 400 to 700 nanometer. In this wavelength range, it comes blue and the red wavelength of light. So there will be active photosynthesis rate. So answer to this will be 1. Both assertion and region are correct, and region is the correct explanation of assertion. In the blue and the red region of spectrum, there is maximum absorption by chlorophyll A. That is why it has a high rate of 104. Read the following 4 statements A, B, C, and D and select the right option having the correct statement. Okay. The jet scheme of light reaction take place in presence of PS1 only. Jet scheme means it should related to PS1 as well as PS2. Wrong. Only PS1 is functional in cyclic photophosphorylation. This is correct. When only PS1 is active, then it leads to cyclic. Cyclic photophosphorylation results into synthesis of ATP and NADPH. This should be wrong. If it is cyclic photophosphorylation, then it should synthesize only ATP. NADPH2 will be synthesized when in the case of non-cyclic. Okay, so this is wrong. Stroma lamella lacks PS2 as well as NADP. This is correct. Okay, the stroma lamella lacks PS2. PS2 is present in the case of grana, but in the stroma lamella, PS2 is absent. Okay. Lacks PS2 as well as Na. Answer to this will be C. Is correct, and then is and chloroplasts without grana are found in. This should be bundle sheet of C4 plants without grana. In the case of C4 plants, they have leaf anatomy which is called as a Kranz anatomy. So in the Kranz anatomy, in case of Kranz anatomy, there will be mesophyll cells, larger number of mesophyll cells, and several layer of bundle sheet cells. Okay, so mesophyll cell will surround the bundle sheet cells. So inside the bundle sheet, the chloroplasts which are present, the chloroplast will not have grana. That is why since chloroplast is absent, that is why it does not have grana. That is why it is called as agranal chloroplast. In case of bundle sheet, the chloroplast does not have grana. It's called as agranal chloroplast. And since grana is absent, PS2 will also be absent. Okay. In the case of C4 plants, mesophyll cells is mainly concerned with light reaction. Mesophyll cell is mainly concerned with light reaction in case of C4 cells, and dark reaction mainly occur in the bundle sheet cell. Okay, so light reactions जो होता है, it mainly occurs in the mesophyll cell, and in the bundle sheet cell it mainly occurs dark reaction. And the chloroplasts present in the bundle sheet cell, they are agranal, and so PS2 is absent. And in case of C4 plants, that is why mesophyll cell is specialized for light reaction and bundle sheet cell is specialized for dark reaction. Kelvin cycle will occur in the bundle sheet cell, means C3 cycle will occur in the bundle sheet cell, and in the mesophyll cell it will have C4 cycle. Okay, so here it will have C4 cycle, and here it will have C3 cycle. So in the C3 cycle, glucose will be synthesized, but in the C4 cycle, organic acid, means oxaloacetic acid, will be synthesized. And in mesophyll cell, light reaction are specialized, and in case of bundle sheet cells, dark reaction are specialized. So chloroplasts without grana are found in bundle sheet cells of C4 plants. Which is, chloroplasts are found in the case of bundle sheet of C4 plants, which is related to light reaction of photosynthesis. So photosynthesis light reaction is related to PS1, light reaction center is P, photolysis is associated, this should be wrong, photolysis is associated with PS2, and PS1 and PS2 are associated in dead skin. This is correct. In PS1, light reaction center is P680. This should be P700. And PS2, the light reaction center is P what? Okay. So this is given ulta. So PS1 and PS2 are associated in dead skin. Oh. Which one of the following is correct? Photorespiration is a wasteful process. This we know because ATP is consumed but glucose is not synthesized in the case of photorespiration. Photorespiration is absent in C3 plants. Photorespiration is present in C3 plants. C3 plants are more efficient than C4 plants. This should be, this is given ulta. That means answer should be this. C4 plants are more efficient than plants. C4 plants are more efficient than C3 plants. Why? Because C4 plants does not have photorespiration, a waste process, and C4 plants also has more amount of chloroplast, and because of trans anatomy, the fixation of carbon dioxide is also more efficient. That is why C4 plants are more efficient, and they are also related to tolerance of high temperature. इसलिए C4 plants जो है, they are more efficient than C3 plants. Okay. Next is question number 108, which is false about C4 pathway. Primary carbon dioxide acceptor in E is, this should be is, PEP. This is correct. Enzyme for carbon dioxide fixation is peptidase. Peptidase is monofunctional enzyme and it has capacity to fix only carbon dioxide. Mesophyll cell lack rubiscope. Mesophyll cell lack rubiscope. Correct. Answer should be bundle sheet cells contain peptidase. In the case of C4 pathway, carbon dioxide acceptor should be PEP, but bundle sheet cells in case of C4 plants should have rubisco. Peptidase is found in the case of mesophyll cell cytoplasm. Answer is 2. This is wrong. Bundle cells in pappcase. Pappcase should be in the mesophyll cell. 109. Stroma lamellae have both PS1 and PS2, but the membrane of grana lacks PS2 as well as NAD reductase. Stroma lamellae have both PS1 and PS2. Okay, this is wrong. But the membrane of grana lack PS2 as well as NADP. This is wrong. Membrane of grana should have PS2 as well as NADP reductase. But here it is written membrane of grana lacks. It should have. Okay, means this is wrong. Cyclic photophosphorylation also occur when only light of wavelength beyond 618 nanometer is available for. This is correct. Cyclic photophosphorylation जो है, it should occur only in the presence of PS1 के presence में ही होना चाहिए, and also occur when only light of, it should occur only in the presence of PS1 and beyond 680 nanometer, means 700, P700. So assertion is not correct. Assertion is not correct, but reason is correct. 4. 110. Which statement about photosynthesis are correct? First carbon dioxide acceptor in C4 cycle is PGA. This should not be PGA. In C4 cycles, the acceptor should be what? PEP, phosphenol pyruvate. In C3 plants, first stable product of photosynthesis is RU. This is not RUBP, it should be PA, phosphoglyceric acid. Stable product in C3 plants should be PGA. PGA is 3 carbon. That is why it is called as a C3 cycle. Cyclic photophosphorylation results in formation of ATP. Correct? Oxygen liberated during photosynthesis comes from water. That means C and D alone are correct. इसमें ऑप्शन जो है C and D alone आ रहा है, ये दोनों ऑप्शन सेम आ रहा है, ये चेंज होना चाहिए. It should be B and C. ये आंसर should be this. C and D is correct. In photosynthesis, P680 refers to reaction center of photosystem 2. अगर photosystem होगा 1 होगा, then that should be P700. But here it is P680. P680 means it should be photosystem 2. In a chloroplast, the highest number of protons are found in chloroplast, highest number of protons are found in lumen of thylakoid. Why? Due to the process of electron transfer system. Okay, matched column 1 with 2 and found the correct option. So column 1 में दिया है C3 plant, then column 2 में these are the product. Okay. So primary carbon dioxide acceptor, in C3 plant it should be RUBP. Correct. Carbon dioxide acceptor is RUBP. Okay. Primary carbon dioxide fixation product. Primary carbon dioxide fixation product should be PGA. C3 cycle में PGA. Okay. Cell type in which the Kelvin cycle take place. In C3 plants, Kelvin cycle will take place in mesophyll only. So C will be 3. Mesophyll only. And cell type in which initial carboxylation reaction with respect to C3 plant, that also will be mesophyll cell. So answer to this will be A2, B1, C3, B3. A2, B, A1, okay, A1, B2, C3, D. A1, B. In the case of the C3 plants, there is only one-time fixation of carbon dioxide. Rubisco is a bifunctional enzyme. Rubisco will fix the carbon dioxide, and RUBP is the acceptor, and the stable product form is PGA. And there is only one-time carbon dioxide fixation in the case of Calvin cycle. And all the fixation of carbon dioxide will occur in the mesophyll cell only in the case of C3 plants. So A1 answer is 4. A1, B2, C3, and D will also be 3. Okay. With reference to factors affecting the rate of photosynthesis, which of the following statement is not correct? Means incorrect option. Okay, increasing atmospheric carbon dioxide concentration up to 0.05 can enhance carbon dioxide fixation. This is direct NCERT line. Presently in the atmosphere, the carbon dioxide concentration is about 0.3 to 0.04% according to NCERT. But if this carbon dioxide concentration increases to 0.05, then it will be good for plants with respect to carbon dioxide fixation. This statement is correct. C3 plants respond to higher temperature with enhanced photosynthesis, while this is wrong. Actually, C4 plants, they should not be C3 plants, it should be C4 plants. C4 plants respond to higher temperature with respect to C3 plants. This is wrong. Answer is this. Tomato is a greenhouse crop which can be grown in carbon dioxide enriched in atmosphere for higher yield. This is correct. If it is a greenhouse crop, that means generally greenhouse crop are C3 plants. So they'll be kept in a greenhouse so that sunlight intensity is reduced and carbon dioxide are being trapped. So this is correct. Light saturation for carbon dioxide fixation occurs at 10% of full sunlight. This is also correct. So answer should be this option 2. For PEP, phosphenol pyruvate is primary acceptor of carbon dioxide in the case of 4 plants. The first stable product of carbon dioxide fixation in sorghum. Sorghum is C4. According to NCERT, some example of the C4 plant are given like maize, sorghum, sugarcane, atriplex, like this. So this type of plants only will be given in the option. Either it will be given maize, sorghum you'll find in previous year's question. Sugarcane is the plant in which C4 cycle was been discovered. Okay, so stable product of carbon dioxide fixation in sorghum is, it should be oxalic acetic acid. First stable product form. Oxaloacetic acid is 4 carbon compound. That is why only C4, it is called as a C4 cycle. Stable product is oxaloacetic acid. NADP reductus is located on the stroma site of the membrane of thylakoid. Okay, this is correct. Inside the chloroplast, it will have, inside the chloroplast, it will have thylakoid which stack one on top of another. Okay, and this NADP reductase enzyme is located here. Located here. That means it is located on the grana but towards the stroma side. This is stroma. So it is located on the grana towards the stroma side. Correct? During proton accumulation in human, there is a measurable decrease in the pH in the stroma. This should be increased because all the protons will be, because of ETS, initially before ETS, there is a proton concentration here. That means it was acidic. But because of ETS, the proton are pumped inside the lumen of thylakoid. So proton gradient will increase inside the lumen of thylakoid. So it says here, during proton accumulation in the lumen, there is a measurable decrease in the pH in the stroma. So when all the proton will pump inside into lumen of thylakoid, here the pH should increase. It is given a decrease, so it should be as it is entered inside pH 2, it should be increased. So this is wrong, but this is correct. Assertion is correct, but reason is not correct. Assertion is correct, but reason is not correct. Which of the following is not true regarding the release of energy during ATP synthesis through chemiosmosis? From previous year, this is a previous year question. This type of question they are asking repeatedly. Okay, so let's see. It involves, it should be not true, that means which is incorrect. Movement of proton across the membrane to the stroma, this is correct. Because of ETS, proton will pump inside into the lumen of thylakoid. Reduction of NADP to NADPH2, this will occur in non-cyclic photophosphorylation. Breakdown of proton gradient will occur inside the lumen of thylakoid. Proton concentration increases, that proton will again pass through CF, not CF1, and synthesize ATP. So breakdown of proton gradient will occur, but breakdown of electron gradient, this will not occur. Okay. So little bit in brief, I will say this part. Like in process of ETS, because of ETS, in the beginning, initially there was a proton concentration here, but because of the ETS, all the proton has pumped inside into the lumen of thylakoid. So there is a high proton gradient in the lumen of thylakoid. तो यहाँ पे CF naught CF1 रहता है. So CF naught, the structure is like this. CF naught CF1. Its another name is ATP synthesis, जो कि attached रहेगा in the thylakoid. So through this, when proton will pass through it, whenever 3 proton will pump through it, it will synthesize ATP. In some book, it is given 2 protons. In some book, it is given 3 protons. In some book, it is given 4 protons. Okay. So I am following this 3 proton because it will also apply in the case of mitochondria. So in some book, it is given 4 protons also. In some, it is given 2 proton also. Okay. So this, whenever 3 proton will pump through this CF naught CF1, it will synthesize 1 ATP. So this high proton gradient, which is present in lumen of thylakoid, when passed through this CF naught CF1 and synthesize ATP, this whole process only is called as the chemiosmotic hypothesis, which was given by scientist P. Mitchell. So proton concentration increases inside the lumen of thylakoid because of ETS, and that proton gradient, when it breaks down, then it synthesizes ATP. So proton gradient should break down, and this is a form of facilitated diffusion, because higher concentration to lower concentration, it is a form of passive transport, जो कि अंदर से high concentration जो हुआ है वो break down करेगा and comes out through that F naught F1 CF naught CF1. जब ये proton pass होता है inside this CF naught CF1, a conformational change occur inside this ATP synthesis. CF1 CF naught is a part of enzyme ATP synthesis. Okay, this all we have done in class. So CF naught CF1 is a part of enzyme ATP synthesis. When proton pass through it, it brings a conformational change in this CF naught CF1 and leads to synthesis of ATP. So this is answer. What is the role of large bundle seed cell found around the vascular bundle in T4 plants? This should be to increase the number of chloroplasts for the operation of Calvin cycle. Okay. In the case of T4 plant, they have a Kranz anatomy. Mesophyll is undifferentiated. It is of one type, and bundle seed is of several layers. So inside the bundle seed, rubisco is present, and rubisco is a bifunctional enzyme. whenever carbon dioxide is high, Rubisco will fix carbon dioxide. Oxygen high, Rubisco will fix oxygen. So because of that, Rubisco should get a high amount of carbon dioxide only, that since it is present inside the bundle sheet. So because of that, in the case of the C4 plant, in order that to increase the number of chloroplasts for the operation of Kelvin cycle, because Kelvin cycle is operated by what? Rubisco. Rubisco is a bifunctional enzyme. So Rubisco should get higher number of carbon dioxide. So inside the C4 plants, inside the bundle sheet, more amount of chloroplast is there, and it will have more amount of carbon dioxide inside it. Carbon dioxide concentration will be more since chloroplast is also more. And from mesophyll, malic acid will be transported to bundle sheet, and mesophyll, little bit here I'll explain here in brief for all of you. C4 plants में क्या होता है? Carbon dioxide is fixed from the environment, और ये bundle sheet है. So inside the bundle sheet, here chloroplast will be there. So carbon dioxide is fixed 2 times in the case of C4 plants. Peptidase enzyme will fix it, and it will form OAA. In brief, only I'm saying, and it will convert to form malic acid. Malic acid is transported to bundle sheet. Malic acid undergo decarboxylation inside the bundle sheet, and after decarboxylation, carbon dioxide will be released inside a bundle sheet. So Kelvin cycle will perform here. Kelvin cycle is performed by Rubisco. So since more amount of carbon dioxide will come here, Rubisco will get only carbon dioxide. Okay, so that is why this option is correct. To increase number of chloroplasts for the operation of Kelvin cycle, more amount of chloroplasts will be. Okay, so that Rubisco get only carbon dioxide. Given below are 2 statements. The primary carbon dioxide acceptor in C4 plant is PEP and is found in mesophyll. And is found in mesophyll, correct? Mesophyll cells of C4 plants lack Rubisco enzyme. Mesophyll cell of C4 plant lack Rubisco enzyme. This is also correct. Mesophyll cell of C4 plants will have only C4 plants will have only peptidase. It does not have Rubisco. And in the statement 1, this is correct. The primary carbon dioxide acceptor in C4 plant is phosphonol pyruvate and is found in the mesophyll cell. Both statement 1 and 2 are correct. Identify the correct statement regarding chemiosmotic hypothesis. Splitting of the water molecule takes place on the inner side of the membrane, means inside the lumen of thylakoid. Inner side means towards the lumen of thylakoid. This is correct. Proton accumulate within the lumen of thylakoid. This is also correct. Primary acceptor of electron electron transfers the, it should be comma here, transfers the electron to an electron carrier. Not be electron carrier, this should be hydrogen carrier. Hydrogen carrier means actually, what is it? Be plastoquinone. Plastoquinone can carry both electron as well as proton. NADP reductase enzyme is located on the stroma site of the membrane. It is located on the thylakoid, but towards the stroma site. Correct? Proton increase in number in stroma. The answer should be A, B, D. A, B, and D. 122. Productivity and yield are better in C4 plants. Productivity and yield are better in C4 plant. Why? Because photorespiration is absent and occur at a very low rate, and more number of chloroplasts is also there, and they have higher tolerance to temperature. These are the region. C4 plants lack photorespiration. Also correct. Productivity and yield are better in C4 plant. Why? Because they lack photorespiration and they have more chloroplast and they can tolerate temperature. These are the region. So assertion and region are correct, and region is the correct expression of assertion. 1. Glycolysis electrons are removed by, it should be electrons are removed by NAD ion. In the process of glycolysis, NAD ion gets the electron. The second step, second half of glycolysis, PGAL undergo oxidation and convert to form 1,3-phosphoglyceric acid. The second half of glycolysis. So PGL undergo oxidation. This is the only oxidation step in glycolysis. So PGL will undergo oxidized, and the electron which is released from PGAL is received by NAD ion to form NADH2. I write it generally NADH2, or in NCERT it is given NADH plus H. Okay. So electron is obtained by NAD ion. And what is NAD ion? NAD ion is the form of coenzyme which is present inside the cytoplasm of cell as well as inside the matrix of mitochondria. This is a form of coenzyme. So electrons should be removed by NAD ion. Chemiosmotic theory of ATP synthesis in chloroplast mitochondria is on basis of, this is proton gradient. Enzyme of TCA which is located in inner mitochondrial membrane eukaryote and cytosol in prokaryote. This is a very simple question. Succinate dehydrogenase enzyme. Overall goal of glycolysis, Krebs cycle and electron transport system, there should be ATP in a small stepwise unit. 27. C4 plants show tolerance to higher temperature. This is correct. C4 plants have both Rubisco and Pepcase enzyme. This is correct. C4 plants have Rubisco where in bundle sheet and Pepcase where in mesophyll cytoplasm. So they have both. They have Rubisco in the bundle sheet and they have Pepcase in the mesophyll, and they show higher tolerance to higher temperature. This is so correct. So answer should be both statement 1 and 1. Pyruvate dehydrogenase complex needed for conversion of pyruvic acid to acetyl, it should be 2 acetyl coenzyme A is located in, that should be fixed of mitochondria. Pyruvate dehydrogenase complex, what does it do? It breaks down pyruvate to acetyl coenzyme A. That process is a linking reaction between glycolysis and Krebs cycle, and it will occur inside the matrix of mitochondria. Which step of Krebs cycle carbon dioxide is evolved? That should be oxalosuccinic acid to ketoglutarate acid. Linked reaction between glycolysis and Krebs cycle is, just now I said, there should be pyruvic acid to acetyl coenzyme A. It is intermediated by enzyme pyruvate dehydrogenase complex. 131. Dark reactions are not directly light driven. Correct? They are not directly light driven, and dark reaction occur in darkness. This is wrong. So statement 1 is correct, but statement 2 is incorrect. See, dark reaction are not directly light driven. They are not directly light driven means they are light independent. That is why it is called as dark reaction. It is light independent. But dark reaction occur in darkness. This is not related. Means dark reaction means which is carbon dioxide fixation. Okay. which can occur by C3 plant or which can occur by C3 cycle or C4 cycle. So this dark reaction, it depends on a type of plant. In the case of C4 plants, C4 cycle as well as a C3 cycle, this will occur during the daytime. C4 cycle, C3 cycle, which is a dark reaction, it will occur in the daytime. But there's a different type of plant which is called as the CM plant, which is not in the NCERT, but it is in the case of CM plants, C4 cycle will occur nighttime, C3 cycle will occur in the daytime. So it depends on a different type of plant. So that is why dark reactions occur in darkness. This is a wrong statement because it also can occur in the daytime also. After glycolysis, fate of glucose in mitochondrial matrix is glucose fate in mitochondrial matrix is after glycolysis. Glycolysis means what? Glucose to pyruvate. So here question is asking after glycolysis fate of glucose. After glycolysis fate of glucose, what will it do? It will form pyruvate, and from pyruvate it will undergo what? Oxidative decarboxylation. And oxidative decarboxylation will form, what will it form? Acetyl coenzyme A. Okay. And this acetyl coenzyme A will start the Krebs cycle. So this fate of pyruvate will be, it will perform oxidative decarboxylation and form acetyl coenzyme A, and acetyl coenzyme A will perform the pathway of Krebs cycle. So pyruvate to acetyl coenzyme A is a linking reaction. Fate of glucose after glycolysis will be oxidative decarboxylation. In which of the following process carbon dioxide is not released? Active fermentation does not release carbon dioxide. What is the role of NAD ion in cellular respiration? It functions as an electron carrier. NAD ion, which is a coenzyme, it is a coenzyme, but inside the process of respiration, it functions as an electron carrier. Like NAD ion will receive the electron from glycolysis or from Krebs cycle. It will carry that electron and will again oxidize in the process of ETS to create the proton gradient, and when that proton gradient breaks down, it forms ATP. So initially inside the glycolysis and the Krebs cycle, NADA will receive the electron and it will get the electron and convert to form NADH2, and that NADH2 will be a carrier of the electron, and when this NADH2 will undergo oxidation, it will lead to proton gradient and synthesize ATP. So what is it functioning actually? It is functioning as a source of an electron carrier. It receives the electron and then converts to form NADH2, and that NADH2 get oxidized to convert to ATP again after creating a proton gradient. Which of the following statement is incorrect? This is correct. C cycle are present in mitochondrial matrix. Glycolysis occur in cytosol. Also correct. Glycolysis operates as long as it is supplied with NAD ion that can pick up hydrogen atom as well as electron, which are released in the process of oxidation. PGL undergoes oxidation. In that oxidation, the electron and proton which are released are obtained by NAD ion. Oxidative phosphorylation takes place in inner. This should be the answer. Match column 1 with 2 and choose the correct option. Photolysis. Because of photolysis, oxygen evolution occurs. Phosphorylation means ATP synthesis B4. Phosphoglycolate means it is a product of photorespiration B2. Carboxylation is done by 21. So A3, B4, okay, C2, 1. A3, 4, 2, 1. Answer. Which of the following statement regarding mitochondria is incorrect? Enzyme of electron transport are embedded in outer membrane. Inner membrane. क्या पूछा है? Incorrect. This should be the answer. This is the answer. Inner membrane is convoluted with infolding, that is called as cristae. This is correct. Mitochondrial matrix contains single circular DNA molecule and ribosome. This is also correct. And outer membrane is permeable to monomer of carbohydrate, fat, and protein. This is also correct. Pyruvate dehydrogenase activity. This requires magnesium. 138.1. Again, incorrect option. Role of oxygen in aerobic respiration is limited to abdominal space. Correct. At last, in the process of ETS, electron is given to oxygen. ATP is synthesized through complex 5, means ATP synthesed. Complex 5 means ATP. Is correct. Oxidation reduction reaction produce proton gradient in respiration. These are all correct. In ETC, one molecule of NADH2 give rise to, it should be 3 ATP, and one molecule of FAD should give rise to 2 ATP. Okay. This is given wrong. The number of times decarbonylation of isocitrate occur during single TCA cycle is, this should be 2. The 5-carbon compound formed during TCA cycle is, in the Krebs cycle there are many intermediate or substrate form. Among them, some are 6 carbon, some are 5 carbon, some are 4 carbon. So citric acid, aconitate, oxalosuccinic acid, this all will come under 6 carbon, and alpha ketoglutaric acid will be 5 carbon. Okay, so this is 6 carbon, this 2 will be 4 carbon, and this is the only 5 carbon compound form. 142, glycolysis occur in cytoplasm. This is correct. Enzyme for glycolysis are found in cytoplasm. Correct. It is common in aerobic and anaerobic. Glycolysis is the common respiratory pathway between aerobic and anaerobic respiration, and the enzyme are located in the cytoplasm of the cell. This should be what are correct, and it also explains. 1. Column 2 and choose the correct option. Electron transfer system. Electron transport system, it should be inner mitochondrial membrane. A is 2. Glycolysis occur in the cytoplasm of cell. B1. Aerobic respiration occur within mitochondria. Respiration pathway is amphibolic pathway. Respiration pathway is amphibolic pathway because it involves both catabolic and anabolic. Aerobic respiration, majority of the pathway of aerobic respiration, Krebs cycle, linking, ETS, oxidative phosphorylation, that occur inside the mitochondria. In the case of eukaryote, only glycolysis will occur inside the cytoplasm. ETC will be the mitochondrial membrane, glycolysis will be plasma cell, aerobic respiration will be within mitochondria, respiratory pathway is amphibolic pathway. A2, B1, A2, B1, C3, C4. Rosette habit of cabbage can be changed by the application of gibberellic acid. Rosette means dwarf habit. Like on a stem, if all the leaves are clustered to at one point, like in the case of cabbage, all the leaves will be clustered at one point on a stem, and they will have a small dwarf habit. That kind of habit is called as a rosette habit. So if you apply gibberellin to that, then the stem of the plant will be elongated. So this should be gibberellic acid. Right. Hormone used to speed up molting in the barley, promote flowering in pineapple, and laying leaf senescence are respectively. It should be this gibberellin. Match the column 1 with 2. Light reaction will occur in grana of chloroplast A4. Dark reaction where will occur? 3. Water splitting complex is PS2 1. Trans anatomy should be light reaction A4 A4 3 A4 A4 B3 E1 4 3 E1 2. Okay, so light reaction means it should occur in the grana of chloroplast, correct? Dark reaction means it should occur in the stroma of chloroplast, correct? Water splitting complex is associated with PS2, this is correct. Trans anatomy test. So D should be 2. A4, B3, C1, 2. Which of the following growth hormone regulators known as stress hormone? ABA, abscisic acid stress hormone. Which regulators are used to prepare wheat-free lawn by the gardener? 2,4-dichlorophenoxy acid. In order to increase the yield of sugarcane crop, which of the following plant growth regulators should be spread? If the stem elongation and sweetness is required in sugarcane, then gibberellin is applied, stem elongation. The number of substrate level phosphorylation in 1 turn. Okay. 1 turn of citric acid cycle, it should be 1. Substrate level phosphorylation, 1 turn of citric acid cycle is 1. So thank you.